Okay, it's 1 o'clock now.
Okay.
Maybe we will start the seminar. And before we start, I'll just give you a quick announcement. There is a simultaneous interpretation ready. So if you want to listen to English, you can click English in the bottom. If you speak in Korean, 한국말로 듣고 싶으신 분은 아래 그 초이스에서 영어하고 한국어 중에서 한국어를 선택하시면 되겠습니다. 이제 영어로 좀 얘기를 하겠습니다, 이제. Good afternoon, and welcome to today's webinar on developing an affordable and accessible COVID-19 vaccine where we are now. My name is Chang Nok-seo. I am the president of both Human Asia and the Korean Association for Human Rights Studies, who are co-hosting this event together with Human Asia's student partner group, UAEM Korea. Our speaker today is Dr. Jerome Kim, Director General of International Vaccine Institute. Thank you so much for joining us today. Human Asia is a non-profit human rights organization aiming to create a holistic and inclusive Asia where everyone may live without discrimination. To achieve this mission, Human Asia offers a wide range of educational programs to empower regional human rights activists who are the centers of future human rights movement. Amongst these diverse programs is our Access to Medicines project in collaboration with UAEM Korea. This project seeks to mobilize the network of Access to Medicine students activists in UAEM Korea to promote progressive, patient-centered pharmaceutical and innovation policies through advocacy, research, and campaigns. Our Access to Medicine project only began last year in September. Yet the developments of 2020 and the spread of coronavirus have seen this project take on previously unforeseen dimensions and significance. Regardless of age, gender, socioeconomic, or ethnic background, our right to health is one of our most basic and essential rights. It is therefore a practical and moral imperative that we use every tool at our disposal to develop and deploy a COVID-19 vaccine quickly and on a scale for everyone to have accessibility. Throughout this process, however, and after our scientific communities have successfully developed a vaccine, we must ensure that all involved actors, from governments to pharmaceutical companies to NGOs, strive to continually adopt a human rights-based approach in response to the ongoing global epidemic. Only then will we guarantee that no one is left behind and left out. Lastly, I'd like to express my gratitude to the members of Access to Medicine for your dedicated works, engagements, and activities. It has been so unfortunate that I wasn't able to meet you in person so far. I really hope to see you very soon. Now I'd like to invite Hana Chang, President of UAEM Korea, to say a few words.
Thank you, Dr. So. Hi, I'm Hannah Chang. Thank you so much for being here. I'm the president of UAEM Korea. I know some of you are UAMers, but for some of you, maybe it's your first time to hear about UAM. So UAM stands for Universities Allied for Essential Medicines. It's an international student group for advocating universal access to medicines. Last year, we found the Korean chapter of UAM, and since then, we've been working on our campaigns and research projects. And we're working closely with Human Asia and other organizations. We used to focus on access to orphan drugs in Korea, but with this corona pandemic, we're also having projects on the access to medicines for COVID-19. We believe it's important for us university students to get involved with access to medicines movement. That's because universities do play a role in the drug pipeline. They're mainly involved with basic science research and preclinical studies. So we should be concerned about how these university research is used. There haven't been any student organization in Korea striving for access to medicines. So at UAE Korea, we're trying to raise public awareness and highlight on blind spots in the access to medicines in Korea. We hope university students can get more active in this issue, and so we can work towards our goal, which is universal access to medicines. Thank you.
Thank you very much, Hannah. I will see you very soon. Okay, I'd like to now invite Dr. Jerome Kim to begin his session. Dr. Kim is the Director General of International Vaccine Institute and an international expert on the evaluation and development of vaccines. Dr. Kim was the Principal Deputy and Chief Laboratory of Molecular Biology and Pathogenesis in the US military HIV research program, and also served as project manager for the HIV vaccines and advanced concepts evaluation project management offices, US Army Medical Material Development Activity. He has authored over 250 publications and has provided his expert opinion on both domestic and international news outlets. Thank you very much for coming here, Dr. Kim. I will pass over to you now.
Oh. Okay. Thank you very much, Professor So and Ms. Chang, for the kind introduction and your very thoughtful commentary on the COVID crisis and accessibility to vaccines, which are essential medicines. It's hard to imagine what it was like at the beginning of the new year, 2020. You know, I guess the Chinese government had made an announcement in December 30th of the previous year that there was some sort of outbreak going on in Wuhan. But very few of us could have imagined that we would be in the situation we are today, where billions of people had been locked down, where hundreds of thousands of people have died, and where over 10 million people around the world should be stricken by a new pathogen, one that we had never before imagined would be present. But this is not the first pandemic that the world has faced. In fact, this is a cartoon from 1983, sorry, from the early 2000s, about a pandemic that started in 1981, which was the pandemic that is known as the HIV/AIDS epidemic. And is spread all over the world. Almost 40 million people are infected, and every year, at least 1 million new people become infected. But many of us remember a disease called the bubonic plague, also known as the Black Death, which struck Europe many times in the medieval era, 1562 being one of the more recent episodes of Black Death. And this had a population shaping impact on Europe's population, on its countries, on many of the things and the thinking that went around what was happening and why the Black Death was happening. Smallpox, which has now been eliminated by vaccination, was first described about 3,000 years before the Common Era, and in 1979 was declared eliminated from the face of the earth by the World Health Organization. The Blue Death. No one thinks about cholera anymore, but when cholera first arose in 1817, it caused a huge panic in Europe. They didn't know what to do. This was a disease where a normal and healthy young man could be alive one night and dead in the morning. Cholera has been through 7 global pandemics. We are in the middle of the 7th global pandemic now. A century ago, the world was afflicted with the Spanish flu, which came on just before the end of the First World War, but continued to devastate world populations for a year to come in 2 successive waves. Hence the second wave fears for the current COVID-19 pandemic. And then, of course, we have COVID-19, and this is a scene from a Spanish church illustrating the large number of unburied as churches, mortuaries, hospitals struggle to keep up with the demand for beds and with the demand for mortuary services caused by the COVID-19 pandemic in Europe. So what are coronaviruses? Coronaviruses, oops. Okay, sorry. Coronaviruses are what we call promiscuous, in that a single coronavirus could easily infect members of different mammalian species. So what you see in the middle, here, is are bats. And bats are the source of many of the coronaviruses that have caused problems recently in humans. The original SARS, now known as SARS-CoV-1 outbreak, started in bats, transitioned to palm civets, which are used as food in certain parts of China, and then infected human beings, causing the large outbreak of SARS in 2002. MERS, the Middle East Respiratory Syndrome, is another beta coronavirus that again started in bats, transitioned through an intermediate host in camels, and then infected humans. Luckily for us, infection of humans and human-to-human transmission is not terribly efficient. So MERS, although it has a significant mortality rate approaching 40% in some countries of the world, really hasn't been the cause of a global pandemic. And then we have SARS-CoV-2, which again started in bats, passed through perhaps some other intermediary, spiny anteaters or pangolins have been implicated, although it's not entirely clear that they are the secondary host, and then infecting humans, where after an initial outbreak in Wuhan in China, described in December of 2019, has spread to over 200 countries in the world, infected over 10 million people, and killed over 400,000. Oops, let's see, there we go. So COVID-19, which is a description of the disease, and here you see a 103-year-old COVID-19 survivor. Unfortunately, as people age, as they get above the age of 65, mortality rates escalate very dramatically. But in general, 80% of people who are infected with COVID-19 develop mild or relatively asymptomatic disease. But 15% of people are hospitalized. So if you think about 15% of people being hospitalized in a disease that's affected 10 million people, we are looking at over 1.5 million hospitalizations. Of those, 5% are intubated and 2 to 3% die. And when you consider that in many parts of the world, access to ventilators is very limited. In one country in Africa, there was only a single ventilator in the entire country. You can see the problem that COVID-19 would cause as it begins progressively to move from high-income countries like Korea, the United States, Italy, France, Germany, into poorer parts of the world where access to healthcare, particularly intensive healthcare, are extremely limited. We know that there's age-dependent mortality. Mortality is very low in the young, but can be very substantial in the elderly. We believe that transmission is through respiratory droplets, perhaps through aerosols, perhaps less so through contaminated surfaces. We know that infection-fighting proteins called antibodies are detected in most of the people who have been infected. However, the most important kind of infection-fighting protein, those antibodies that bind to and inactivate the virus, known as neutralizing antibodies, are often high in severe cases, but actually can be fairly low in asymptomatic cases. And this raises a question of whether these people who had asymptomatic disease will truly be protected against the second exposure. The other thing that we found is that people had thought that COVID-19 would infect a country and that we would rapidly come to some equilibrium, what we call herd immunity through natural infection. It turns out that that probably will take longer than we thought, although there's been some dispute recently in the literature about how many people are actually infected. But in one country, Sweden, where herd immunity, where the government decided to not take a lockdown approach, but rather to just use social distancing, mortality rates have been remarkably high, much higher than any of the European neighbors of Sweden. What you see in the little cartoon is a comparison of the major pandemics. And you can see SARS-CoV-2, COVID-19 is actually down on the bottom left. It was still small, and although it's growing and may now be larger than the Ebola outbreak of 2014, 2015, it is still relatively small compared to some of the other major outbreaks that the world has seen. It is, however, the biggest pandemic of our generation, and certainly of the last century. Let's go now to COVID vaccines. Are vaccines truly the way to get from the Brooklyn Bridge, while New York was locked down, to the Brooklyn Bridge of the past? So the bridge back to normalcy or normality. Is the COVID vaccine going to be that bridge? When you're developing a vaccine, the first thing you do is you think about what you don't know. The first and probably the most important question is, does infection provide protection against reinfection or immunity from reinfection? And why is that question important? Well, for a classic vaccine disease model, and I'll use polio as an example, although the disease has many manifestations, a person who acquires the polio virus could be asymptomatic or mildly symptomatic, maybe with diarrhea, or they can be a person who'll develop paralysis and death. So the disease is is different. The manifestations are very diverse. However, everybody recovers from polio. The body's immune response, your protective response, develops infection-fighting proteins or antibodies, killer cells, which kill virally infected cells. And this immune response initially controls and then eliminates the virus, and also subsequently protects you against reinfection. And that is really important. Why? Because when we develop a vaccine, we want the vaccine to do exactly the same thing. When we inject a person with the vaccine, we're training the body's immune or defensive system to recognize that virus, to develop strong protective immune responses against the virus, so that if the virus ever appears, or if the person is ever exposed to the virus, those same immune responses, which target the virus very specifically, will be rapidly able to eliminate the virus without causing disease or infection. The second, and so for those diseases for which vaccines have been developed, that classic model is usually the case, that the body is able to contain the infection and eventually eliminate it and then protect against reinfection. For diseases like HIV or TB, we know that the body's defensive system cannot eliminate the infection. So a person who is infected with HIV never recovers spontaneously. A person infected with tuberculosis is infected for the rest of his or her life, and the disease can reactivate. We know that as well. And so because the body's immune system is unable to contain or eliminate the pathogen, it's much more difficult to develop a vaccine that will do the same thing. The next question is, which of the immune responses is most important? Is it the infection-fighting proteins, or is it the killer cells? Now, with HIV, for instance, we think it's the infection-fighting proteins. With COVID, we think it's the infection-fighting proteins, the antibody against COVID-19. With tuberculosis, many scientists believe that it is the T cells, those cells that recognize and destroy the TB bacillus, that is the most important part of immune response to tuberculosis. But again, we often don't know the answer to this question completely. Oops. The third question is, what is the most appropriate animal model? Because vaccine development is a long and complex process and very costly. So it really helps to be able to look at experimental vaccines in animals to see if the concept works, to really see, is this vaccine developing the correct protective responses that we think are important. So what you see on the right is a monkey holding a rat. And it's often said for vaccine development, mice lie, monkeys exaggerate, only humans tell the truth. And what does that mean? It means that often when we look at the immune responses in a mouse, we see great responses to the vaccine. When we vaccinate the monkeys, the responses are less, but we sometimes see protection. But when we take that vaccine into humans, sometimes we really don't see what we were expecting from the monkey model or from the mouse model. And so it's very important to choose the model correctly. And for HIV, for instance, every single monkey study that has suggested that a vaccine will work has been wrong. And that is exactly the problem we don't want to face with COVID-19 because of the rapid spread of infection, the significant number of hospitalizations, and the huge number of deaths. And the final question, and probably one of the most important questions that vaccine developers think about is the vaccine safe? Actually, what you see in the picture is a reaction to the introduction of the original smallpox vaccine. So smallpox was eliminated, was wiped off the face of the earth by vaccination with another virus called cowpox. And so when cowpox vaccination was introduced for smallpox in the 19th century, so this is the 1800s, some people mistakenly believed that the introduction of, quote, cowpox would cause cows to appear on their arms, and that's what the cartoon is showing. Of course, we know that that's not correct, and in fact, that same cowpox vaccine caused a disease, an infectious disease, to be wiped out. And we would like to do that for more infectious diseases. This is the typical development pathway for a vaccine. Usually, it takes 5 to 10 years. The cost, between 500 million to 1.5 billion US dollars. The failure rate, that is the time from the laboratory to the license to sell the vaccine, the failure rate is 93%, so that only 1 in 10 vaccines make it from the lab to licensure. That's a huge failure rate and a very significant cost, so is it any wonder that companies take their time in the development process? A vaccine goes from its initial stage in the laboratory to testing in animals, that's called the preclinical stage, to phase 1 trials in humans. Phase 1 trials look at whether the vaccine is safe. They may look a little to see if the vaccine develops the correct immune responses or protective responses, but really it's around safety, and it's usually about 50 or less people. Phase 2 involves hundreds of people and is really the first look at the ability of the vaccine to generate the correct protective or immune responses, and again, in a larger number of people to ensure that the vaccine is safe. Phase 3 studies look at both efficacy. Does the vaccine work to prevent infection or disease? And is the vaccine safe in thousands of people? A study that I did in Thailand involved 16,402 Thai men and women, volunteers. Another study that was done in Thailand involved 40,000 Thai school children. Again, these are very important studies. They're very large, very complex, and quite expensive. So is it any wonder that the companies move through these stages very slowly, very deliberately, to make sure that as they move from one stage to the other, that they are relatively assured that this vaccine is going to be a success? But what are we planning to do with COVID-19? And you see that up here. We are planning, instead of 5 to 10 years, making this vaccine in 6 to 18 months. So all of the steps that were listed below have to be accelerated and compressed. And how are we going to do that? This is a 5-year cycle. What you see on the left in green, preclinical stage, testing the vaccine in animals. We then move into phase 1. At the conclusion of phase 1, usually after about 12 or 18 months, we can analyze the safety data from volunteers who receive the vaccine. If everything looks fine, then we move into phase 2. Phase 2 also lasts usually about a year, maybe a year and a half. And at the end of that, we know now with increased certainty that the vaccine is safe, and also that it's making the correct protective or immune responses in what we call the target population, the group of people who will get the vaccine in the end. Assured of that, then a company moves to phase 3, which is the very large test of safety and efficacy. And that usually takes 2, 3, or 4 years, depending on the number of volunteers enrolled in the study and the number of cases of the infectious disease that appear during the time of the study. At the end of that phase 3, hopefully, the company will have shown that the vaccine protects against infection and disease. And then they submit, usually on the order of 100,000 pieces of paper, to a regulatory agency like the Ministry of Food and Drug Safety in Korea or the Food and Drug Administration in the United States. That organization usually has a year to review the dossier. And at the end of that time says, we agree with you, this is safe and effective, it should be marketed, or they disagree. And companies really don't like when they've gone all the way through phase 3, when they see a signal for efficacy, and then they're turned down. So usually, by the time it gets to the regulatory agency, we're pretty certain that a vaccine is safe and effective. So what are we going to do for COVID-19? So you see that now on the bottom. We start phase 1 often before the preclinical work is complete. A month after the final dose in phase 1, so if it's a 2-dose vaccine, you give it at month times 0, and then 1 month later, and then 4 weeks later, you analyze the safety data in 40 to 50 people. It looks like it's safe, then we move immediately on to phase 3. We don't complete phase 1. We continue to follow the patients, but we start enrolling the second phase. So several hundred patients now in phase 2. 2 months after the final dose in phase 2, so again, maybe 2 months or 3 months into the study itself, we analyze them quickly to see if the right protective responses are being made and whether the vaccine is safe. And if we find that it's immunogenic and safe, then we move on into phase 3, which is the test of efficacy and safety. And phase 3 trials are usually designed with multiple looks to make sure that the vaccine isn't hurting people, but also to see in an interim analysis. So these are formal statistical analyses to check in the interim whether the vaccine is showing really, really significant efficacy, prevention of infection and disease. Because if it is, it may be considered unethical to continue a vaccine trial if the vaccine is really working. On the other hand, if the vaccine is causing harm, it's also very important to stop the study for ethical reasons, again, for the safety of the volunteers. So assuming that at month 3 or month 4 in the phase 3, we find that the vaccine is highly effective in preventing infection and disease, we will probably continue the study, but then report that to the Ministry of Food and Drug Safety or the FDA and ask for licensure for the vaccine to be approved. And rather than have a year to review all the data, the Food and Drug Administration, the regulatory authority will have a few weeks, because the pressure of the epidemic has caused regulatory agencies to really look carefully at what they consider to be really critical information. So you can see, oops. Sorry, it's not, there we go. That we can shorten the clinical trials period, and we're still looking at the same efficacy signal, whether the vaccine works. But what we really don't have is the longer-term safety follow-up. So in the usual sense, we have 5 years to collect safety information and long-term safety information. In the truncated protocol for approving a drug, a vaccine in COVID-19, we're going to collect information for maybe a year before the vaccine is approved and ready for use in humans. Now, is that a big concern? Probably not that large a concern, because most safety side effects, most safety signals that are seen in the first 4 weeks to 8 weeks after a vaccine is given. But there have been cases of safety signals arising after a vaccine was actually approved. So for instance, the Sanofi dengue vaccine was approved, and the safety signal that was seen and eventually caused a reissuing, a revision of the original recommendations for use appeared at year 3. As the immune responses to the vaccination were dropping, they saw a potential safety signal. So the company revised the recommendations for use. So sometimes, even with the normal process, we see problems that develop years on, and we have to be ready. So it's going to be very important that as we look at COVID-19 vaccines, that we remember that we have to complete the safety analysis, and we have to follow volunteers who receive this vaccine for an extended period of time. The World Health Organization has come up with a set of recommended guidelines for vaccines. What's shown in the far right panel are the minimum standards for WHO approval, and then the preferred standards. So this is what we would take this is what we would want. And you can see here the measure of efficacy. So the vaccine has to be 50% effective in preventing infection or disease as a minimal criterion, and as a preferred criterion, at least 70% effective in preventing infection or disease. So again, the World Health Organization has come down with one thing. The US FDA has just issued a draft guidance, again, saying that 50% is the minimum criteria for vaccine efficacy if a company would like to be licensed in the United States. So what are the vaccines? And I'm going to go through this briefly. This is a cartoon of the virus. What you see in blue are what we call the spike proteins. This is an expanded view of the spike protein. It's what we call a homotrimer. In pink is the receptor binding domain. The antibodies that block virus entry into cells target the spike protein, and in particular, the receptor binding domain. So if we were really good, what we want is a vaccine to generate antibodies against this, but in particularly against the RBD, or receptor binding domain. What kind of vaccines are being used? Nucleic acid vaccines, like RNA or DNA. Recombinant protein vaccines, like the kind that are used for certain of the hepatitis vaccines. These are just a piece, just the spike protein itself, usually mixed with something called an adjuvant to strengthen immune responses against this particular protein. A lot of the vaccines that have moved forward very quickly are what we call vectored vaccines. We take a piece of the genetic information of the SARS-CoV that encodes this spike protein, and we insert it into a different virus. And the three candidates that are moving forward have this piece inserted into an adenovirus, a type 5 adenovirus for CanSino, a type 26 adenovirus for Johnson & Johnson, and a chimpanzee adenovirus for AstraZeneca and Oxford University. Inactivated vaccines are very easy to make, and they're among the most commonly used and also the cheapest of the vaccines that we can develop. In that case, you just grow the vaccine up and kill it with heat, formalin, or another chemical substance, ultraviolet radiation sometimes. And finally, we have live attenuated vaccines. These might take much longer to develop, but if they're effective, are much more effective as vaccines. What you see on the far right is a panel, a table, taken from Wikipedia, because I found that Wikipedia updates their sources more quickly than everyone else. But you can see the number of different vaccines out of a total of 194 that have been reported to be potential candidates. Really remarkable, very quick progress. We think now that there are 17 or 18 vaccines in human clinical trials. Again, given the fact that until January 11th, we really didn't even know the sequence of the virus, to now have 17 or 18 vaccines in clinical trials and 194 being worked on is really unprecedented. This gives you a better idea of what I mean. So Moderna was the first company to have a vaccine in humans. They started their trial March 16th, 2020. The first data were available from Moderna at the end of April, and they expect to have vaccine available for vaccination in vaccine programs by October. You can see the other companies listed here. This was as of June 9th, 2020. How many are there now? A lot more. Again, 17 or 18 according to the last World Health Organization assessment. So really very rapid progress, but you can see here that many of the companies really are expecting to have vaccine available for use by the end of this year or beginning of next year. So people often ask, why is there no vaccine for MERS or SARS-1, but we expect to see one for COVID? And this is an illustration, and it's actually a very useful illustration. This is the SARS outbreak, the Ebola outbreak, the Zika outbreak. The timing of the outbreak and the time it takes to develop a vaccine is shown in yellow. So typically what happens is you have an outbreak, it gets into the news, governments get worried and they put money into it, and then a number of different organizations, industry, biotechs, universities start to work on vaccines. And usually by the time the epidemic is tailing off, the first vaccines come out. So here you see the end, so the end of the epidemic, the end of the money, the end of the project, and there is no vaccine developed. And that's exactly what happened to SARS and MERS. With SARS, there was a huge outbreak. People started to work on vaccines. SARS-1 disappeared. The Chinese shut down the sale of palm civets, and we've never seen it again. It is 80% similar to SARS-CoV-2, so we in essence are seeing the cousin of SARS-1, but SARS itself disappeared, never to reappear. The SARS-1 vaccines are all on the shelf, not being used. With MERS, the outbreaks tend to be very small, so by the time we get around to making a vaccine, the outbreak is gone. So if you had started to work on a vaccine when the MERS outbreak took place in Korea, by the time you had a vaccine to be tested in animals, the outbreak was over. And for a company, switching to production of a new vaccine is very disruptive. They have to take resources away from things that are really important to the company financially. And what they found is that they couldn't interrupt their normal activity to just take up whatever new outbreak tended to happen. And this is what happened in Ebola. And it took President Obama calling the vaccine manufacturers to say, we really think that this is important. We want you to work on an Ebola vaccine. And they dropped everything. They went to work on Ebola vaccines. And then the epidemic ended. And only one company was able to take it all the way to completion. The Ebola vaccine is not a vaccine that the company will be able to sell for a profit. So if you're a company that just spent hundreds of millions of dollars to develop a vaccine, stopped other projects that would potentially make money for the company, you can see why companies are very reluctant to get involved in work on pandemic pathogens. What's the difference with SARS-CoV-2? Well, one of them is that this virus appears to be lasting a bit longer. So here it is in January. We are now at in July. We're at 10 million cases, 500,000 deaths. This is not going away. So there is adequate time to develop a vaccine, even if we're looking at a 12 to 18 month time frame. There have been a huge number of deaths, and this is from the United States. Their first wave never ended, but they're in the middle of another peak of virus-related cases. And we know with COVID-19, the impact is huge. The impact on global GDP so far is 8.4 trillion US dollars. This is the drop in economic activity in the major economies that occurred as a result of SARS-CoV-2. And we're still not recovered yet. So how are we going to make a difference? Well, the US government announced, we call it OWS, Operation Warp Speed. This is the Starship Enterprise traveling at warp speed. But the basic point here is that time, impact, and money will make the difference for COVID-19. And what do we mean by that? The US government, for instance, and this is just a single government, has put $3 billion and made $3 billion available to companies to accelerate vaccine development. AstraZeneca has gotten $1.2 billion. Johnson & Johnson, $456 million. The RNA vaccine company, Moderna, $430 million. Down at the bottom, Emergent, Ology, and SiO2 Material Science, $782 million. What does the presence of money do? Money de-risks a project. So if you're thinking you're a vaccine company, you have a 93% failure rate, you're very reluctant to take things forward because you're very worried you're going to fail. But what has the US government done? It said, okay, money is no object. We will pay you to develop this vaccine. Not only that, but the US government has paid certain companies to begin manufacturing of the vaccine when they're in phase 3, before we even know that the vaccine works, on the hope that when the vaccine works, we will have vaccine made and available for use in the United States, not necessarily anywhere else. So taking that time frame and the failure rate and the cost, the US government's $3 billion acknowledges the risk and attempts to mitigate it. That's how we're going to accelerate vaccine development for COVID-19. There have been, and I'm going to go through this kind of quickly, there have been several pieces of evidence that a vaccine will be possible. One of the things, again, that we use very commonly to see if something will work in humans are monkey studies. So far, there have been 4 non-human primate studies where they vaccinated with a candidate vaccine and shown that the monkeys are protected against infection or disease or both. With the Sinovac, which is a Chinese company, a whole inactivated vaccine, the higher dose of vaccine, 6 micrograms, is associated with a significant reduction in the amount of virus present in the throat and almost no detectable virus in the lung. So again, this is a vaccine that would protect a monkey against infection. There's a good, not perfect chance that it would protect a human. Jenner, is a chimpanzee adenovirus vaccine. It doesn't do as well at preventing this kind of infection. In fact, you still see a small amount of virus in the lung, but compared to the 3 monkeys that got placebo, it's significant reduction in the amount of virus and the amount of visible disease seen in lung biopsies. So again, this is the Chadox vaccine. Other companies like Inovio, which has a DNA vaccine, do not yet have monkey data, but they have evidence of production of these neutralizing antibodies in mice. And some hope then that when the monkey challenge studies are done, that they will be able to show protection against challenge in monkeys. But right now, for Inovio, only mouse data and production of neutralizing antibody. There is one company, CanSino, that has reported data from humans in a major paper in The Lancet from their phase 1/2 initial study in humans. The disappointing part of this, so CanSino's vaccine is actually put into a viral vector called adenovirus type 5. The problem with adenovirus type 5 is that many of us, most of us in Korea, but many people around the world have already been exposed to adenovirus. So unfortunately, what the immune system sees is not the SARS-CoV-2 piece, the spike protein, but it sees instead adenovirus, and it goes crazy. And so at the medium dose group here, only 50% of the people who receive the CanSino vaccine develop the neutralizing antibodies, the antibodies that inactivate the virus. In the high dose group, it was 75%, which is much better and would be a reasonable vaccine. However, in that highest dose group, 40% of people had fever, and 14% of them had the highest level of fever, grade 3 fever. So the company has had to use the medium dose, where only 50% of people develop neutralizing antibody. Will it work in the end? We're not sure, because only the efficacy, the phase 3 trial, will tell us. With this pandemic in particular, we've had a problem with people not publishing their data, but reporting it in press releases. And this is data from Moderna, which is the RNA vaccine supported by the US NIH. They reported results in their human volunteers, their phase 1 volunteers, and they reported that they saw neutralizing antibodies, the antibodies that inactivate the virus, in 8 out of 45 volunteers. Was that because they tested 45 and only 8 were positive? We don't know. We think that there's more data coming, and that in fact they only looked at 8 people, and in those 8 people, all of them had neutralizing antibodies. But what they don't tell us in this press release is how high those neutralizing antibodies were. In fact, they say, well, they were the same as you would see after a COVID-19 infection in a human. Well, what we know is that in some humans, especially those with asymptomatic or mildly symptomatic disease, sometimes we can't even detect neutralizing antibody. So releases like this really end up only confusing the situation and making it difficult to interpret whether or how serious or how good the vaccine will be. So this is about IVI. Very briefly, we're an international organization, 35 member countries, and now Finland and Madagascar are pending final submission and are hoping to join, we hope, this year. Oops. We work all over the world, Latin America, Sub-Saharan Africa, South and Southeast Asia. We do everything related to vaccine work, from epidemiology to making vaccines in our own laboratory, to testing them with funding from Gates or Korea, Sweden, or India, helping companies license their vaccines, and then using those vaccines in effectiveness trials to generate real-world evidence. How are we different from some of the other organizations like CEPI, Gavi, the Global Fund? You can see IVI works across the spectrum as an executor of activity, whereas these organizations fund activity in particular areas, usually after vaccines are licensed for Unitate in the Global Fund, and Gavi really only after a stockpile is authorized and created. So what is IVI doing for COVID-19? We've decided not to make our own vaccine, but to help companies like Sumagen, Genexin, GI Innovation, and Genofocus develop their vaccines. Now, there are other companies with whom IVI has applied for funding, and they're not listed here. And until we are certain that funding will be available, we won't list them. But really, our goal is to accelerate vaccine development in any way we can. So here you can see that Genexin's DNA vaccine is actually undergoing human clinical trials at Yonsei University. IVI is also working with Inovio we're hoping that this will start sometime in early July to test their DNA vaccine in Korea with Seoul National University Hospital and the KNIH. We've received some funding from Sweden to initiate surveillance activities in sub-Saharan Africa and from Germany to work in South and Southeast Asia. Now, what are the concerns around SARS-CoV vaccine development? You know, there's been a lot of what we call vaccine nationalism, and what IVI and many other groups, especially those that work internationally, feel is that this vaccine, what we're fighting for is a vaccine for everybody. We're really looking for a vaccine that will be able to be used in the almost 8 billion people who inhabit the planet Earth. We've seen a huge amount of misinformation, usually provided over the internet, but also stoked to some extent by scientists who are prizing urgency over objectivity and are willing to publish unfiltered science or make press releases rather than provide data for other scientists to review. There are a lot of COVID conspiracy theories, like one that says that Bill Gates only invented the epidemic so that he could inject microchips into everybody, which is, you know, completely false. But there are people who believe that. This has stoked the anti-science. One of the things that those of us who work in HIV vaccines remember is the push by the groups most affected by HIV to accelerate science. Because in the end, it was science that was going to develop the highly active antiretroviral therapy or an HIV vaccine. With COVID-19, we're seeing the rise of anti-science and people opposed to any kind of vaccination, even if it's a vaccine that will prevent them from becoming infected. What we see mixed in here are politics, particularly in countries that are having difficulty controlling the outbreak. Miscommunication, economic hardship, partisanship over something which should be open, international, where scientific progress should be made and where public and global health policy should be transparent and decisive. A potential safety issue is something that we are aware of. With the original SARS-1 outbreak, whole inactivated virus vaccines were used to vaccinate mice. And in those mice, the vaccine did what it was supposed to do. It lowered the amount of virus present after challenge, which is great. But what we saw also was that in some of the mice that were protected, these particular kinds of cells called eosinophils were infiltrating the lungs of the mice. This did not occur in every case. And in fact, it didn't occur sometimes if you used a different species. It didn't occur sometimes if you used exactly the same system, but without the adjuvant. Again, sometimes it was very difficult to tell why we were seeing this. It was also seen occasionally with the MERS vaccine, again, raising the possibility that this might be a coronavirus-specific phenomenon, that although we saw protection, we also saw this problem with secondary infiltration of the lungs with eosinophils. And so this summarizes that question. What is it about SARS-CoV-1 or MERS that was associated with this odd eosinophilic infiltrate? Was it the animal model? Was it the type of vaccine? Or was it the adjuvants that were used? Of note now 4 monkey studies of COVID-19 vaccines show no evidence of the enhanced disease. 3 whole inactivated virus vaccines and 1 chimpanzee adenovirus vectored vaccine have been shown to protect the monkeys, and then also shown not to be associated with evidence of eosinophilic infiltrate. So again, we hope that this will mean that when we test these vaccines in humans, that we again will not see any evidence of the secondary infiltration of eosinophils. So COVID-19 vaccines have not shown a safety signal in monkeys, but we must still ensure that human safety information is collected completely and accurately. Finally, I'm going to go into access, and this is really about access. All projects that were funded by the Coalition for Epidemic Preparedness Innovations, which has about $1 billion to work on COVID-related vaccines, have signed a global access agreement that ensures that countries around the world will have access to the vaccine at an affordable price. Importantly, President Xi of China also announced that China's COVID vaccines will be available for global public good. Now, what does that mean? We're not exactly sure, but what it told us was, that at least for China and Chinese vaccines, that China was going to work very hard to ensure that the world could access at an affordable price vaccines for COVID-19. So what you see in the world map on the bottom in green are countries that have implemented rotavirus vaccination. Over 95 countries around the world have put rotavirus vaccine into their national vaccination programs, which is a great thing. However, you can see a big gap here in Asia. Less than 20% of Asian countries have implemented rotavirus vaccination programs. Rotavirus vaccine is really remarkable. It was licensed in 2007 in the United States. Almost after a few years, rotavirus vaccination rates in the United States were at 70%. At that point, rotavirus diarrhea disappeared from the United States. It's very difficult to find a child in the US now who has rotavirus-associated diarrhea. That's how good the vaccine is. WHO approved the vaccine in 2009. In 2014, only 20% of children around the world had received 3 doses. Now, this is despite the fact that rotavirus vaccine is in the Gavi stockpile. Gavi makes the vaccine available for roughly $2 a dose, which is much less than the $100 a dose that parents in the United States pay for the same vaccine. Yet, only 20% of countries have implemented fully rotavirus vaccine programs for the children present in those countries. This gap in access, 70% versus 20%, is after 13 years, is exactly what we want to avoid with COVID-19. When a COVID-19 vaccine is available, we want that vaccine to be used quickly and effectively in countries around the world that need it. So COVID-19 vaccine development, to summarize, vaccine development conceptually is simple. 3 things: prove the vaccine works, make the vaccine, and use the vaccine. The caveats, though, are very important. We have to prove the vaccine works and is safe over both the short term and the long term. We have to make the vaccine in quantity, with quality, and affordable. We have to use the vaccine to reduce the burden of COVID-19 with respect to access and equity. So if we're going to go from the Spanish steps in Rome after COVID to the way the Spanish steps in Rome were before COVID, we're going back to the future. So first, finally, some thoughts. Can we prove that a vaccine works and is safe? This is a prediction. Yes, I think we can, but can we do that in 12 to 18 months? Cautiously optimistic if everything works as planned. And I mean, unfortunately for vaccine development, not everything works as planned. Second, can we make the vaccine in sufficient quantity at high quality and affordable cost? Perhaps. CEPI, the Coalition for Epidemic Preparedness Innovation, has identified global manufacturing capacity in excess of 10 billion doses a year and has secured 2 billion glass vials for vaccines. You will not believe this, but there is a global glass shortage that is preventing us from having enough vials for vaccines. This is the kind of thing that where COVID-19 affects logistical capability and where the demand for vaccines and vaccine vials is exceeding the current supply. CEPI believes that it will be able to make 2 billion doses of a safe and effective COVID vaccine by the end of 2021 and will distribute it around the world according to an access framework that CEPI and the United Nations are developing, with contribution, of course, from the Global Alliance for Vaccines and Immunization, or Gavi. Several countries around the world, including Singapore, Korea, and others, are really working hard to make sure that vaccine nationalism does not rule the day and that populations around the world that are in need of the vaccine will have access. So these are the predictions. And if you're a baseball fan or a physicist, one of these two actually said this: it's tough to make predictions, especially about the future, but we can hope that we will have a vaccine, that it will be available in sufficient quantity, and that we will have access and equity at the end of the day. Thank you.
Thank you very much, Dr. Kim, for your highly insightful and informative presentation. We'll now open the floor to questions from our audience. First question was about clinical studies. So the question was, by what criteria are monkeys selected as an appropriate animal model?
So So I think I gave the joke that vaccinologists use, which actually has a significant grain of truth, which is that mice lie, monkeys exaggerate, and only humans tell the truth. And why do I say that? Because if we were when we use mice as the model, we often see very strong immune responses. In particular, mice are bred into certain strains, certain strains really, really overemphasize the production of these infection-fighting proteins. If you go into more complex mammals, so into larger mammals like rabbits, you see usually often a decrease in the immune response. When you get to monkeys, sometimes what was very immunogenic in mice is hardly creates an immune response in monkeys. And when you go into humans with the same vaccine, you see nothing. So monkeys are closer to humans in terms of their physiology and their immune system. And so that when we see things in monkeys, we are more assured that we will see something important in humans. And then the same applies for the adjuvants, the things we use to boost immune responses. Often we can't see a difference in mice. If we do see a difference in monkeys, then we're pretty sure that we will see the same effect in humans. The other part of it is that you can challenge a monkey. So with COVID-19, in the beginning, it wasn't possible to challenge a mouse. So you could vaccinate a mouse, you could look at protective responses, but we couldn't infect the mouse and show that the vaccine prevented infection. Now with certain genetically modified mice, that might be possible. Importantly, in monkeys, you can do that. You can vaccinate them, you can infect them, and you can show that the vaccine prevents infection. When you infect a monkey, also, the pathology that is seen looks very similar to what is seen in humans. Most of them develop very mild disease, but they do get the same kind of lung disease that humans have when they develop COVID-19 infection. So again, as a model, it tells us really what we need to know in terms of protecting those crucial organs in humans that are affected by COVID-19. I hope that answers the question.
Thank you so much. And we have another question about virus mutation. What are the chances of having mutant strains of SARS-CoV-2? In terms of vaccine development, how should it prepare for virus mutation?
That is a great question. We have a vaccinologist being developed now as we speak. That's a great question. So what we do know about mutation in SARS-CoV-2 is that it is tenfold lower than we see in influenza and is millions-fold lower than we see in HIV. So, around the world up to April, there were 11 circulating strains of COVID-19. In a single HIV-infected human, so a single HIV virus infects a human subject. That's in 80% of the time. It's a single virus. Within 2 weeks, that virus has generated millions, if not billions, of unique forms, recombination, point mutations, multiple point mutations, so that, in an HIV-infected person, often viruses within the same person can differ by as much as 15%, which is really remarkable. SARS-CoV is on the far end of that. It mutates much, much less rapidly. Are we seeing strains that are different? Yes. Can we characterize outbreaks, for instance, in San Francisco or somewhere else in the world, and examine the unique characteristics of an outbreak, say, in Queens, New York? Yes, we can. Because we can look at minor genetic differences. The question is, does that create a separate immunological strain so that the vaccine won't work? And right now, we don't know the answer to that question. There's one in particular that people are worried about. This is a strain that appeared in Europe. It's called G614D. That refers to the specific change in the amino acid. And what happened was initially in Europe, it was mainly viruses from China. But then at some point, this G614D took over and now is responsible for most of the cases in Europe. It hasn't made the same jump to the United States, but people are expecting that it might. What's unique about it? It may not be that it's so different immunologically, but it is different in its ability to grow and multiply in humans. So there's some thinking that it spreads more readily, which explains why it's taken over the outbreak in Europe. Will the vaccines work against it? At this point, we don't know. And so, for instance, our lab and other labs are looking to see whether the plasma from SARS-CoV infected people from Korea or from Europe will inactivate this virus. And right now, we don't have the data yet.
Thank you for your answer. We have another question about accessibility. So what do you think is the biggest obstacle to development of SARS-CoV-2 vaccines? out of virus mutation or insufficient government support or etc. What kind of steps do you think should be taken under the current situation to accelerate the vaccine development?
So we're already moving faster than we have ever moved for the development of a new vaccine. There are very few vaccines that are developed in less than 4 years. 5 to 10 being the average that we normally see for vaccine development. We're talking about developing this vaccine, about having a vaccine proven to work by the end of the year, when we really didn't know about this virus at the end of last year. And that is unprecedented speed. And we're already pushing the limits of what some people would consider to be acceptable. We are moving from phase 1 to phase 2 without having full safety information. We're moving from phase 2 to phase 3 without having full knowledge that the vaccines are actually making the correct protective responses. We're trying as much as possible to get the vaccine out as quickly as possible, but using in the very end, the very important criterion that the vaccine has to protect against infection or disease and has to be safe to the point where we can, to know, which is about 12 months worth of safety information. To make it faster than that would be very difficult.
Oh, we have many more questions, but since we're running out of time, I think we should be wrapping up. Thank you again, Dr. Kim, for sharing your time and expertise with us all today. I hope that you all remain interested in UN Korea's and Human Asia's future activities and continually engage with our work promoting the right to health for all, both in Korea and across Asia. Thank you so much.
Thank you, Ms. Chung. And we are very committed to access and equity for all vaccines, but right now, particularly for COVID-19.